everyone. Welcome to today's live broadcast, Novel Precision Medicines That Exploit Proximity by Dr. James Prudent. I'm Christina Jewell of Labroots, and I'll be your moderator for today's event. Today's educational seminar is presented by Labroots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many questions as we can. You can enlarge the slide window by clicking the screen icon in the lower right-hand corner of the slide window. Now, if you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button on the lower left. I would now like to introduce today's speaker, Dr. James Prudent. Currently, Dr. James Prudent serves as the Chief Executive Officer and Chairman of the Board at Centros, a biotechnology company focused on developing precision therapies for diseases with high unmet need. While exploring interesting natural products, Dr. Prudent and his colleagues discovered that the all-powerful sodium-potassium ATPase was interacting with multiple proteins at the cell surface. The biology of the various interacting proteins indicated an evolutionary pattern of energy regulation coupled to growth, motility, and survival. This discovery led to the invention of EDC technology, which allows for the unprecedented precise delivery of drugs. Currently, the company has discovered multiple EDCs for cancer and autoimmune disease. Prior to Centros, Dr. Prudent served as Chief Science Officer and board member at Aerogen Biosciences, now Luminex, where he invented gene code and multi-code technologies that incorporate an expanded genetic code into real-time and multiplex diagnostic assays. Before Aerogen, Dr. Prudent invented Invader technology, while at Third Wave, now Prologic. Invader is complementary to PCR, but does not amplify DNA, making Invader more accurate and easier to use. Today, Invader is being used by Hologic and Exact Sciences in their diagnostic products. Prior to Third Wave, Dr. Prudent received his PhD in chemistry at the UC Berkeley under the direction of Professor Peter G. Schultz, and his BS in bacteriology and medical microbiology at the University of Wisconsin-Madison. He has over 100 publications, patents, and patent applications. He's a recipient of the Frost & Sullivan Product of the Year Award and Biotechnology Industry Organization Innovation Award. I will now turn it over to Dr. James Prudent for his presentation. Dr. Prudent?
Thank you, Christina, for that great introduction. I'll just step right in and get started here. I'm the CEO at Centros, and I'm going to give you a little bit of background on exactly what we do here and what we found. We go to the second slide here. We kind of think about delivering drugs similarly to how a postman actually brings his drugs, or excuse me, his packages, his envelopes, his even the FedEx man brings the packages right to a zip code in a particular city or town. This is actually pretty precise if you think about it. The world is a pretty large place, but a few numbers can bring that particular package right to where you want it. I was just showing two maybe not so precise mailmen here, but you can get the point. Antibodies are basically the same type of delivery system when we talk about antibody drug conjugates. So if we look at this slide, we see that the mailman is the actual antibody, and the package is the small molecule drug which is attached to it. If we go to the next slide, the first step in an ADC's activity is really to have that antibody or mailman actually bring the package to the house or the cell. And that cell could be most any type of disease cell that the antibody can recognize. If we then look at the next step of an ADC is actually the internalization, which is basically bringing the mailman into the house along with the package. And then the next step is actually dropping the package off. And in this case, the package really isn't all that precise because, you know, it could be going to a number of people that live within the house or the business. So there's a name on it, typically in a package, but not so much in an ADC. The next step is that package to actually do its job, so whatever it is, it has to act. In an ADC case, it's a drug acting on either tubulin or DNA. So these 4 steps are quite complicated, and they have some deficiencies. For example, the antibody can only recognize a single protein, and that protein may actually be on the surface of normal cells or cells that you don't want to destroy. The second step of internalization is actually a very slow and cumbersome problem. You can imagine if the mailman had to get a signature or actually enter the house with a large package, it's not as easy as dropping it into a mailbox. If you look at 3, the next step is the actual releasing of the package. And in an ADC case, that means that the linker or the connection of the drug to the antibody actually has got to break down or be modified so that that drug can actually get off of the antibody. And so you can imagine that then all of a sudden that drug can be floating around and actually get out of the cell or target other particular molecules in the cell that may be toxic. And then the 4th is really the drug binding, so the concentration has to be high, and if that drug does get out of the cell, which there are pumps that actually take it out, you can build up resistance. So we asked the question if we could build a next generation of molecules where there's only one step. On the left, you see that this would maybe be no internalization, no breakdown, which would lead to novel mechanisms that you'd have to find, and actually you could actually go after new targets. Otherwise, you could think about antibodies that are targeting proteins that don't get into the cell. You could actually have antibodies that don't need to get into the cell. So this would be a factor-acting, more stable, very specific type of system if those 2 targets, the drug target and the antibody target, were actually in close proximity. So I'm going to introduce what we do here. It's called Extracellular Drug Conjugates, and really what these do is they exploit the idea that there are multiple proteins that actually form complexes on the cell surface. And if one of them can be a druggable target, then the antibody can actually be very focused on bringing it to the cell, but only when those 2 proteins are in close proximity, and hence maybe a disease state, you would actually get activity. And so it brings an extra level of specificity to the system. So if we look at what it has to be to actually drive this type of activity, you actually are thinking about proximity effects. And the idea of proximity is really, really fundamental in the areas of science. But I just want to first just talk about looking at this on a real molecular level. So on the bottom left, we show that there's a target cell. And in this case, we fluorescently label 2 proteins that interact. In blue, you can actually see the nucleus of the cell. And then on the surface, the ring around that nucleus is either a green dye or a red dye. And then when they come in close contact, they actually form a yellow color. This is called fretting, but it actually represents, in this case, 2 proteins that do come in close proximity to one another. So if we go up to the top left, we see the 2 complex proteins in this little bitty area that I have marked. And then if we get even closer, we can start actually looking at the idea of a package now in the mailbox, not so much having to get into the house, just putting it into the mailbox. And now that that package is in the mailbox, the actual drug is actually meaning it's in the pocket, and it can actually inhibit or act on whatever target you're interested in. So this is the idea of the extracellular drug conjugate, and I'm going to go into some detail explaining what we've actually determined is very useful for and what targets we've gone after. So if we look at the similarities first between the ADC and the EDC, these are all things that a company needs to think about. Does the antibody or the extracellular drug conjugate allow you to make it, package it, administer it, and have all of the FDA requirements that an ADC does? And in these cases, yes, it does. Now the differences are what I kind of talked about before. The drug location is different. The EDC, it's outside the cell. The linker requirements are different. We never want the drug to leave the antibody. But there's got to be a distance that you have to span between those two proteins that are in close proximity. And then, obviously, they have to be in close proximity to activate. The internalization is not required. It doesn't require breakdown. And in our case, these EDCs kill by a different process than apoptosis. We found that they kill by a system called pyroptosis, which is basically blowing up of the cells and not releasing a lot of their contents that can lead to some sort of immune activation, which in some cases may be good, but too much can be bad. Now, the antibody examples are interesting. Herceptin and Rituximab, two huge blockbuster drugs. ADCs cannot use Rituximab, and EDCs have been found not to be very useful with Herceptin. So you open up an area of a new set of protein targets and antibodies. Now, how do the EDCs that we use work today? Well, basically, we're targeting a protein that we call the all-powerful sodium ATPase. And the reason why we call it the all-powerful, because if you build up too much sodium in the cells will die. And so what we've done is we've targeted with a natural product, a actual molecule that's made by mammals as well as plants called cardiac glycosides or steroidal glycosides, but basically a steroid ring that has an additional enoline attached, and they become extremely powerful inhibitors of this sodium-potassium ATPase, which uses more ATP than any other protein in your body. So it seems to be quite an important protein just for that fact alone. You see the antibody on the top, and then the linker, you could see a little better in the blow-up where it's that light blue that connects the antibody to the drug. And that has to be a certain distance, so it can span the distance between where the antibody has the bound linker all the way down to where the site of the drug is. If we go to the next cell, this is giving back to what I said about the actual EDC blowing up the cell. On the top is really a fluorescently labeled EDC bound to the cell, and you can see it's not internalizing. But at the bottom right, you can see after addition of the EDC, the cells start to balloon up. And the simple fact of the matter is that sodium builds up inside the cell and things go haywire. And so the cells become very, very ill. Dyes can now get in, so that's where the green dye is, and the cells die. This is all published in our paper in Nature Molecular Therapy if you'd like to go to it and read in more detail. Now, do EDCs kill cancer is really the question, and this, I think, is, at least in the mouse model, we're seeing a very nice correlative agreement with as we load in more and more of the EDC, we see more and more reduction of tumor growth over time. And then are they safe? And I'm going to show you a little more about this in the monkey studies. But in general, what we're trying to do, like with every ADC, is to take a drug, make it more toxic because you're going to load more into the target cell of interest, through that delivering it with the mailman or the antibody. And at the same time, we're keeping it away from cells that you don't want to kill because they don't form the complexes, in our case, on the cell surface. And so you see that that yellow line is opening up that therapeutic index by about 1,000-fold just by putting the antibody on. Now, there are three portions to an EDC: the drug, the linker, and the antibody. This is very similar to an ADC. And I'm going to go through each one of these and how they're different or the same as with an ADC. So first, the drug. So on this slide, slide 20, you could see that we've actually done a lot of work on the SAR of this molecule, really looking at all the little functions of what these particular drugs do. And we've modified one so that it not only can be attached to the antibody, but also it's extremely active and not a substrate for a number of different enzymes that could break it down in the body. So we look at drug mechanism then, as I mentioned, we're blocking the sodium ATPase. Sodium removal is required for all cell growth. Interestingly is that sodium comes in a number of different ways, and that's why it builds up so quickly. For example, waste products that have to come out of the cell actually bring in sodium as a co-transporter. So does glucose, but it comes into the cell and brings in a sodium with it. I put a couple other small molecules that actually use sodium to transport without energy. So as this sodium builds up, your body ends up using a lot of sodium just to pump that extra sodium out. And so ATP gets used up quite quickly, and so there needs to be more and more nutrients, especially in diseases like cancer or even autoimmune disease where your immune system becomes activated to your own self. So the drug binding then, if we look into close molecular structure of it, you can see on the top left is our molecule, which has the 6-membered ring pointing right to the purple circle, which is a potassium, and locks it in place. That doesn't allow now sodium to get out or potassium to get in. This is all work done by Paul Neeson, our advisor here at Centros. Now the linker. So on 24, you'll see that this data is quite intriguing because on the top, we're changing the length of the linker while it's attached to the drug and the antibody. And as we make that linker longer and longer, we see more and more activity. In fact, when the linker is very small, we see very little activity. Now, on the bottom, we said, well, what if the antibody got degraded and the drug got released? We actually know now that on the bottom, where in the first case, where we don't have a linker at all, the drug's quite active. But as we add the linker and then increase its length, it becomes less and less active. So this sort of is the key ingredient to this concept, where some people would worry about the drug on an antibody floating around with a target outside of the cell, that these would be rather toxic. But the fact that you have that long linker and the proximity that the antibody places it makes that drug linker conjugate very, very active once it binds to the protein complex on the cell surface. Now the antibody. The first antibody we looked at was a protein called Dysadherin. It turns out to be a known protein that interacts with the sodium ATPase. And it's also a target that is considered a metastatic target prognosticator for metastatic cancers. There's a load of other data on this, and I suggest that if you're interested to go look at this Cancer Letters paper that was published in 2007. But in general, this is a very, very nice marker for a number of proteins important in cancer, and seems to be upregulated quite significantly in metastatic types of cancer. Now the next target is EDC, is our EDC8, which targets CD38, and many of you may know that are scientists in the oncology field. Is CD38 is the target for daratumumab that's been showing some very nice activity in the actual clinical trials, as well as now it's prescribed for people with multiple myeloma. CD38 is a ribose hydroxylase, but it also has some very interesting connections to cancer that I had mentioned at the bottom. It too seemed to be quite closely associated, at least complex, on the cell surface to sodium potassium ATPase, especially when these cells especially when the cells are in a cancerous state. And then I'm going to talk a little about CD20, which is the target for Rituximab. And of course, we all know that Rituximab is an excellent drug, but it also has some fallbacks where the fact that it works through a CDC and ADCC activity makes it a problem with certain patients where they become resistant to it. And then, of course, we have the double hit lymphoma targets, which is the most aggressive form of non-Hodgkin's lymphoma. And I talk a little about the bottom of why cardiac glycosides and inhibiting the sodium ATPase have been shown to be very good drugs for these three targets, c-MYC, MCL1, as well as BCL2. And so I put down some papers that you can go and look at a later time. Now, if we look at the precision, on this slide I'm showing EDC1, but the chart at the bottom shows the EC50 values. Higher bar means that these are more active drugs. They have a lower EC50 value. And then on the right, you see drugs that, you see this cardiac EDC that has very little activity. These are all based on different cell types that express disadherin. So on the left, you see disadherin expression is high. You see activity high. On the right, where disadherin is low, or if cells are in a normal state, you see that the EDC activity is low. In no cases have we ever seen good activity for EDC1 if the cells are not expressing disadherin. Now, the question then is do we really know what the targets are? And so what we did is some knockdown experiments. You can see on the top right in the purple is that if we knock down the alpha-1 subunit of the sodium ATPase, which is really the key target in cancer cells, we see that the activity of our EDC actually goes up. Now, we can't knock down all the ATPase, so we only knock down about 90 to 95%, so the cells still grow. And you can actually see you need less EDC to kill those cells, but the EDCs become really potent when you do. And that's because you have to have a certain number of ATPases to survive, and the less you have in the cell, the less EDC we need. Now, if you knock out disadherin, you can see on the bottom right that that line is flat-lined, meaning no cells are dying because disadherin is not on the cell surface any longer. So then if we go to the EDC8 case, which targets CD38, many of you may know that CD38 can be induced to be expressed in the cell surface by low levels of ATRA. And ATRA is a used compound for certain types of diseases, and it seems to be quite safe in humans. But what's interesting, you can see from the charts on the bottom, is that if we take a cell line that doesn't express CD38 without adding ATRA, you see basically no activity. And then all of a sudden on the right, we made a number of different antibodies that bound to different areas or epitopes on the CD38. And in this case, all of those are now active when they're made into an EDC, or in this case EDC and EDC8, and they become extremely active now that ATRA has been added and CD38 has been expressed. So then the question is what's the toxicity of the parts? In this case, we used our EDC9, which is the Rituximab antibody. And this chart basically shows that the EDC in red in the middle chart is the most active, followed by the drug itself. And then you see way off onto the right, you see the drug linker, and then the drug linker with Rituximab or Rituximab alone. At the bottom, you could see an actual graph of the different molecules that are presented in the middle EC50 curve data. So in general, EDC9 is quite active. A control conjugate or control EDC, which doesn't target, in fact in this case it targets CD47, which doesn't interact on the cell surface but is expressed, is very inactive even when you add it in combination with rituximab. So then what is the efficacy of these EDCs? So first I want to show EDC1. This again targets Dysadherin. And you can see in the non-Hodgkin's lymphoma model you get very nice activity and complete regression. In some cases you get cures. In the middle you see in the middle and the right you see two drug-resistant strains. One is ASPC1 cell line, and the other is a PDAC model, or a patient-derived pancreatic cancer model, where the tumors were resected directly from the patient and then placed into immunoincompetent mice. And in this case, we combined it with Gemzar, and you can see that not only did we get regression, but we also got some cures in these animals, where the two alone were not very active. So it's showing there's a synergy between Gemzar as well as EDC1, and it's quite significant in both cases. In this case, we're looking at EDC8 now, targeting CD38, and one of the in this case, this is a Ramos cell line. One of the strong drugs that's used in the clinic today is called CHOP. And we see that over this particular span of time, we're seeing that very large tumors in the mice that were dosed on day 18. This is a very aggressive tumor. We actually see most of the mice dying before you see any of the tumors starting to grow out in the EDC8 case. So this again is showing that this particular conjugate is quite active in immuno-incompetent mice, even better than CHOP, a pretty aggressive toxic treatment. So then what do we see for EDC9? And in this case we're comparing rituximab directly, and you're seeing that with a single dose of either rituximab or EDC9, we're seeing very good tumor regression in the EDC9 case. In fact, many of these animals survived, and of course there's always the case where some don't, so we're looking at some averages, and we're seeing that the EDC9 is outcompeting rituximab alone. So now, are these drugs safe? And the answer to that is it seems to be, at least in monkeys at this time. So the specificity of this data is showing, this is EDC9, of course, it's going after CD20. This particular drug targets CD20 expressing B cells. So we're looking at CD19, CD20 positive or CD20 positive on the bottom left two graphs. And you can see that immediately these B cells get depleted, and then after about 20 to 30 days, you see that the B cells start to come back normally. Now you look at T cells, natural killer cells, helper T cells, you don't see this kind of a drop. In fact, most of the cases you don't see really any change, besides the fact that these are percentages and B cell levels are going down, so you do see a little increase, but that's based on the percentage of cells in the blood. What's interesting is on this slide, slide 40, you're actually seeing that we can multi-dose these monkeys and actually see the same effect over and over and over again. In the first dose, we see what you saw before. We let the B cells go back to normal. Then we hit them again. We see them drastically get depleted. And then the third dose was a smaller dose, but it was right after the B cells started to come back up, and you can see that they go back down again. Now, we then looked at cytokine response, because clearly you don't want some sort of cytokine storm in this case. And so we compared it to Regeneron's 1979, which is their bispecific. And this is a CD20-CD3 bispecific antibody, and they do see IL-2, interferon T and F levels going up. We did not see those. We actually tested IL-6 as well, and we didn't see IL-6 levels going up appreciably. So this is very interesting and very important as far as we didn't see those kind of cytokine responses that could be worrisome in the clinic. Now, can you produce these? The answer is yes. On this slide, we're showing multiple lots being made. And then what we use is a quick HIC analysis showing that each one of those EDC conjugates that we make are very similar. And at the bottom right, you can see that we're looking at aggregation. In fact, we actually believe in a lot of cases that because of the long linker we use, we see less aggregation. So the antibody conjugates behave quite well and are extremely stable, not only on the shelf, but they also seem very stable in serum. And then you can see the in vivo stability is about on par with most of the ADCs, getting cleared on a half-life of about 3 days. So I just want to recap what I went over. Is one, we have a new generation of antibody drug conjugates that's making their way to the clinic. This looks at protein-protein complexes and uses proximity effects for activity. The precision is basically can be measured by the length of the linker, which is typically around 100 to 200 angstroms in length. These are effective. We're seeing cures in immunocompetent mice, and we're seeing drastic changes in B-cell levels in immunocompetent monkeys. We also believe that they're safe. They're clearly safe in rodents, but they're also safe in primates, cynomonkeys, as well as we don't see the cytokine increase or storm that sometimes antibodies or antibody conjugates can bring. Now, we also showed a robust, reproducible, and long-term stability of these EDCs. So we think we have a nice system, and I think it's going to be interesting to see other types of EDCs coming down the pipeline in the future. So I want to thank you, and Chrissy, I'll give it back to you.
Thank you, Dr. Prudent, for that informative presentation. Now, before we do get started on the Q&A session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. Now, we'll try to answer as many questions as we can, and if we do run out of time, Dr. Prudent will answer questions via email after the presentation. Okay, Dr. Prudent, our first question is, let's go with this one. How do you think they will perform in humans?
$1 question, right? That's $1 million question, and of course we don't know at this point, but that's why we have done so much background work. We hope and we believe that they will be safe, which is our #1 concern with any type of cytotoxin that you attach to an antibody. But in general, if we look at the data we're getting now, we think they're going to be quite safe. The real question, I think, is what is going to be the dosing regimen? What disease will we go after with this? Initially, we're looking at the hardest to treat diseases, but clearly diseases that don't involve high doses and where you just want to remove a certain population of cells. I think it's going to work out pretty well.
Dr. Prudent, how many drugs per antibody are typically needed?
Typically, we only need, and we've done this a number of times, we only need 1, maybe 2 drugs per FAB. And what that means, if we use just the FAB portion of the antibody, just the binding portion, we actually see that we only need 1 drug. So it really, but I think it's really going to depend on the protein complex. Some cases we could probably imagine that there will be multiple ATPases close to a single antibody target. In that case, you probably would want a few more drugs. Typically, we use about 4 drugs per antibody, and that's really a manufacturing issue much more than a scientific chemistry issue.
We have time for one last question. Here we go. Given that the drug target is outside of the cell, why do you think these are so less active on cells that do not have the antibody target?
Yeah, great question. Well, you can see from the data I showed on one of the slides about linkers, and this is discussed a little bit in the manuscript that we published this year. But in general, when we start to add what I call spinach or larger parts of the molecule, like the antibody or the linker, to the small molecule drug, we see less and less activity. And so the question is why do they work anyway? And the thought is that the antibody actually increases the local concentration of the drug to the point where it is always going to be bound to the ATPase during the process by which the antibody binds to its target. So the antibody is bound, the local concentration of that drug becomes extremely high in the molar ratio. So we believe that it's for the majority of the time it's bound in the pocket and inhibiting the ATPase. Where if it wasn't attached to the antibody, it would be circulating in the entire body and would be much less active.
I would like to once again thank Dr. James Prudent for his presentation today. Dr. Prudent, do you have any final comments for our audience?
Well, I just want to thank all of you for joining, listening in, and if you have any questions, please feel free to contact us here at Centros at info@centrospharma.com. We hope that these will someday be a big plus for the set of tools we have to treat diseases, especially for people that are very sick and ill and have the devastating disease of cancer. Thank you again.
And thank you again, Dr. Prudent. I would also like to thank Labroots for making today's educational webcast possible. Now, today's webcast will be available for on-demand viewing through May 23rd, 2017. You will receive an email from Labroots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thanks for joining us, and we hope to see you next time. Goodbye.